I will now go through some technicalities with you, if you allow me. So first, let me check that my screen resolution is good enough. Yes. Okay, that's better. And after which I will go to this tab of my Firefox. I would like to ask you to pay attention even a little bit more than you are doing now. And because we are going to look for the workflow with you, maybe somebody can ping in the chat or just open the chat.
The link is it's already there. The workflow is another PDF in the link that Gleb put up. Presentation 2022 GBI. You will find the presentation as PDF and also the workflow as PDF.
Indeed, thank you very much for that. And if you are completely lost and like clicking, then you can simply go to downloads.microscopy.org, click on presentation 2022. That's quite natural. And GBI, there are both the workshops from yesterday and today. Today, obviously, we have the IDR day, and the presentation is here as the PDF, as Jean-Marie highlighted, and this is the walkthrough. Now, I really want you to find that walkthrough, okay? Because this will indeed give you a hand, and if you don't have it, then stop for a second, ask in a chat or otherwise, say that this was not clear, you can't find this very PDF document. And if you can't find it, then this is the only thing which you should be finding. All the rest is basically inside that walkthrough. So we've done the introduction to IDR, and I will now go through some searching in IDR. Yes.
I think your screen occupies only a portion of the Zoom screen, if that makes sense.
No, it doesn't. Thank you for.
Do you think you can restart the sharing of the screen?
Of course I will.
And restart it. Maybe that will fix it.
Thank you. How about now?
I think it's the same. I don't know if it's just me.
Oh, it's everybody. Oh, it's better
Yeah,
now.
I think.
Okay,
yeah,
so
that's it.
right now I think it's perfect.
Oh, that's what you meant. So this was just the bottom bit of my doc, which was in the way. Okay.
Thank you.
Thank you. Thank you very much for pointing that out. And very good. So we have the first search from publication to IDR study and viewing images and regions of interest. Okay. So let's start with that. Okay, so this is basically documenting what I was talking about in the talk, that you can go via search engines such as PubMed, or going into particular journal search engine, or PMC, which is the one I will now do now. I will right click and open that link in a new tab, or you can simply search Google for PMC in your Google browser, and you should land here in this searching page, which you very possibly know from your scientific career, where you are simply trying to find a journal in some publication from author X. Okay, and my authors X names I know now are Blin, which is the first author of the publication, and then Lowell, which is the last author. It's a publication from Sally Lowell's lab here in Edinburgh. I mean, we are in Dundee, but by here I mean here in Scotland. And if I hit on enter to find that publication, it's the first hit on my on my list. Okay, so this is hopefully not somehow surprising for anybody. I just looked for a publication in a search engine and found that publication. And it's a publication in PLOS Biology from 2019. And of course, I didn't choose those names by chance. I simply know that this publication, the underlying images are published in the IDR. And I can now, first of all, mention that this publication introduces new tools for the automatic segmentation of nuclei within tissues and dense 3D cultures, which are things like mouse blastocysts, which are exactly the images I will be presenting in a second inside IDR. So if I have such publication and I would like to find where the associated data are, I can look for the words data, availability. This is simply a search inside my Firefox, which I'm deploying here on the bottom left. I hope that you can see that. But the data availability statement was actually very easy to find. And it's just right here in this very case. I can click on that and this expands and it says that our imaging data set has been deposited to the image data resource, idr.microscopy.org. This is the basic publication where the IDR was introduced to the scientific community. Under the accession number IDR 0062, so the 62 is the very important bit here. And if I click on this link, it will lead me to the IDR homepage, which you know by now. And if I go there, and remembering that number 62, which I just took from the data availability section of that publication, I can type it in, leaving the search by box here in peace, meaning taking the default suggestion for name, IDR number. This is what I was calling accession number during the presentation, and it's basically 1 unit of submission, which is linked to a scientific publication. I have the autocomplete function here. I accept the suggestion and I have a thumbnail representing the images inside the publication showing mouse blastocyst. Here you can see this is Blin is the first author, and I click on the thumbnail, and from that portal page where I just did my first search, I am navigated to the environment of Omero Web, just as I highlighted in the presentation. Now, the study is here in the tree on the left-hand side, inside something which you might know from Omero world as project, but here we call it experiment. It's the experiment A of this particular study, and here on the right-hand pane, already in the publication, I have the excerpts from this paper. I just researched on the PMC and have other attributes describing also the accession, etc. If I now click on the first, what we call dataset inside that study, I can zoom into the thumbnails in the center pane and select the first image. This is, as I said, the mouse blastocysts, and then I can go to the right-hand pane and see what are the associated metadata. Main metadata are under attributes. So these are the biological metadata from my slide, such as from which organism and cell line the image is coming from, etc. In this case, I would we will go through this harmonica called attributes in many cases later. in the workshop, so I will just leave that in peace now and draw your attention to the attachments, where there is attached a TIFF image, which I can easily download like so into my downloads folder. And you can see that this is a study which deals with segmentation of cell nuclei. And these segmentations were submitted by the authors of the study alongside the original images in the form of a TIFF image, which I'm just now opening locally. And you can see that if I go into that, these are basically binarized images of the segmented nuclei, and you can basically say that they highlight the regions of interest from their image analysis, and thus not only was this image attached inside the database of IDR to the appropriate raw data image, but also the IDR team made the effort to turn the spots in the TIFF images into regions of interest in the Homero sense. Okay, so much so that if I now go and double click on the thumbnail of that image in raw in the center pane to see the raw data, I can, by the way, do this viewing on any image in the IDR. I choose to do so, and I have here, of course, a slider. You can recognize the Homero iViewer, which was presented yesterday in the workshop, and can go through the set sections, etc. And I can switch on and off channels if I wish so, such as I can switch off the yellow channel because I know that it interferes with nice viewing of the regions of interest. And the regions of interest, which I just talked about a second ago, are under the ROIS tab. So when I clicked on that, you could see that the ROIS in the form of masks, which are overlaying the cells inside the blastocyst, appeared. I can show them and hide them by using that button. I can select one of those ROIS or more, and then go to the ROIS menu on the top left and select Export as Table CSV. And if I do so, I can now locally export a rudimentary image analysis from from this particular image, more precisely from this particular ROI, and I have it locally in Excel. And if I wish so, I can now, let's say, compare the analysis with what the authors claim in the paper in the PLOS Biology, which we just studied a second ago. That's their submitted regions of interest. So this is again an important quality assurance step which the scientific community can do inside the IDR. Speaking of which, we can now come back to the study. I just switch the tabs and then let's say go the full circle, where I will find, selecting the top container here, the experiment A, find the DOI data, not the data DOI, but the publication DOI. Data DOI being, of course, this very study inside the IDR, this the data DOI. But the publication DOI, if I click on that, will lead me to the PLOS Biology paper, which I just started on with. Okay. So it's just highlighted in a bit different layout, but you can see this is the study from Blin from Salilova's lab in PLOS biology. And I think if I go to data availability, okay, I will find the IDR, hopefully. It's just right here. I probably made a typo. It's a data availability section. This is basically just going in a full circle from the publication to the IDR and back to the publication. So that is what is possible. And it is, of course, a highly important quality assurance step for the editors of the journal and for the scientific community which wants to study such publications. These images are kind of underwhelming, and they are completely replaced by the IDR team making the effort to put those ROIs into Omero itself by the virtue of turning them into Omero ROIs. Okay, so now when I switched on the Omero ROIs here, the information in those TIFFs, I mean in those TIFFs is completely now absorbed and replaced by these ROIs inside Omero. Now, technically, computationally, these things which I'm clicking on and this TIFF which I downloaded have now nothing to do with each other. If I delete each and every single TIFF from the IDR, it will have no bearing on these ROIs, which will happily survive. Okay, they are simply a different entity. In the moment when Omero team, the OME team put the effort into turning this into that, then it made this image a little bit obsolete. The only thing, the reason for survival of this image and keeping it in the IDR, albeit on a little bit, as I say, not full citizen level, is that these are indeed the original data, processed data, nevertheless original in the sense that they were submitted and produced by the authors of the paper. Okay, so that's why they are there on that attachment level. So as you can see in Omero, you can attach as an attachment whatever file type you like, which we do in this case, and that type file can be an image, but this does not mean that that image is imported into Omero in the full Omero sense, and is thus not a full citizen in the sense, I mean, maybe it's not the best wording, but I don't know how better to put it. It's not an image in Omero sense. It's not an object in the database called image. It's an object in the database called annotation. Okay, file annotation. And Omero doesn't know that it's an image. In other words, okay. So that's the landscape there. Okay, I think that you understand now. Hopefully the others understood as well. It's just a curation decision for highlighting what is important and preserving what is less important, but you know, to be clean. Very good. Let's continue with the second search. Very good. So as the walkthrough says, I will clean my tabs. That's not what walkthrough says, but I will do it anyway. Okay, so I will start on the IDR page, and I will drop down this menu as you saw in the presentation, and I will, pardon me, search for a gene Baz 1A. This is a gene encoding a biodomain adjacent to a zinc finger protein 1A, which is found in some IceY chromatin remodeling complexes. If I if I inspect the autocomplete, the auto suggestion here, I can see that I have two lowercase and uppercase variants, and whether I select any of them, it's the search is insensitive for that. That's by design. So in this case, it always has the nice effect of not missing some search results just because of the lowercasing, uppercasing. In this case, it has biological meaning in the sense that I found both the mouse and human search results. All the searches in IDR are like this with the autocomplete function and case insensitivity. Okay, pardon me for that. So I will go to the bottom of the list and select the lastmost line, which says IDR accession number 0096, which is a publication by Reicher et al. In I can see that in a second. Indeed, if I click on this top container, and you have here the publication title, and again, if I scroll down, as you probably know by now, under the attributes, I should be able to find the publication DOI. I click on that, and quite nicely it opens always in the new tab, which is very sweet because it's very painful to lose the tab where you are. It's published in Genome Research, okay, from 2020, and it's dealing with protein tagging and cellular imaging from Stefan Kubicek lab. Okay. We landed here on this search gave me the results of images associated with the gene BAS1A. The one of those results is inside a study, as I said, by Reicher et al. The other hits are in other studies, obviously, which have nothing to do with this particular paper. And what I'm getting here is, as I say, if I select an image and expand the attributes harmonica on right and pane, I have here under others some interesting metadata, such as the intron targeting sgRNA. I think in your walkthrough there is some funny typo in that part, so forgive me that, which is associated with, of course, the BAS1A gene. And we also observed that the authors have observed a phenotype connected to the BAS1A gene. Okay. The other section is also something which the submitters submit with their images. Now, every image here must be associated with the gene BAS1A. Okay, that's why my search produced them. And if I click on this icon in the gene section, the icon is an external link. The link to the left is an internal link. Okay, this would basically get me into the circle, just like if I would type BAS1A into here. Okay, so I'm not clicking on that because this is an internal IDR link, but I click on that icon and this leads me out into the Ensemble gene bank, and I can read more about BAS1A. And you can indeed see that that is the adjacent biodomain adjacent to zinc finger 1A, and other particularities of that gene and the sequence, of course, etc., etc. The The other metadata I just went through, the study by Reicher et al. Nevertheless contains many more images, not just those few images inside those 5 plates, okay, which would actually come down to 5 images only, yeah, because that's what the number is saying here. It's actually all in plate 5 in the same well, 73, and they are just 5 fields in the same well. That makes very good sense because you can hardly make different conditions for different images in the same well. If you want to see all the images inside that study, you can navigate to this study in the view which we inspected before by going to the right-hand pane, selecting an image, going to the right-hand pane and navigating to a icon saying show parent project and dataset in the tooltip. Okay, this is just the one I'm hovering over. You click on that icon and then obviously the images are contained in plate 5, just like I elaborated on a second ago. And this plate 5 is contained in something which we call screen. The screen is being a container for plates. And this is basically giving you the access to the study itself, okay? I can now go to plate 5 again. In this view, you can see that there are many more, first of all, wells, because we were just in one well. And then inside those wells, there are images. So if I select the well G1, okay, then I can see that I have, first of all, the images contained in that well highlighted, represented by thumbnails in the bottom part of the center pane, and the naming and nomenclature here says fields from well, in this case fields equals image, okay, so one field means one image. The small cartoon here is showing me the positions of the fields alias images inside that well. So if I click on the particular positions in the map, the particular images get highlighted. Again, I can go and study the attributes inside that well. Yeah, again, this is my bus 1A, of course, but now I can, of course, go to other parts of the well. I hope that you understand that this is the high content screening screen plate well format. Where I have, let's say, an automatic microscope and something like a 96 or more well plate, and the automatic microscope shoots an image in each well or more images in each well, and then stores those images inside a format, which is in this case then stored in the submitted to IDR and then imported to Omero as a unit. The images have their own metadata, but if I click on the well itself, this is achieved by clicking on the G1 position here in the middle pane. You can see that I'm on G1 well because it says well ID here in the top right. I get a little bit different metadata, similar but different, and I can click on the tables harmonica here, and I was all the time showing you the attributes, which basically are best of all the metadata which were submitted by the authors with respect to that well. And this is basically then turned into this small table appearing on each well. And then we have a script which will basically pick some of those data and display those metadata in this user-friendly manner under the attributes. So the attributes are, so to say, the best of the tables, which are the exhaustive metadata portfolio for each well. very good. Now, this would be the second type of the search I wanted to show you. Let me just verify I didn't forget about anything. Yes, that's fine. So if there are any questions for this one. Very good. Yeah, I mean the import as plate can have various options and or interesting challenges to face depending on how it is submitted by the authors. And sometimes the curators must do some just to see what I'm talking about. Sometimes the plate is indeed submitted as one unit, as I was suggesting in my speech, by the way I was speaking. But sometimes this one unit comes into fruition only after an effort and kind of interchange between the curators and the submitters, because not all plates are as beautiful as just suggested. So that's the reality. Maybe that's the fair answer to your question in the chat. So basically, that may be a kind of a hint for submitters that the more quirky format and more your format you come up with, the harder it will be to submit to IDR. Whereas the more you comply with the mainstream and the more it is kind of acknowledged proprietary file format, not your own variant, the better you will be faring off during your submission. That's for sure. So this will be for the second search. And the third search, we will go through compound search. Well, compounds meaning some active agents, let's say drugs or otherwise, which are being very typically tested in studies such as these high throughput screening, where I will put in every well some particular compound, and we will concentrate on a compound called remdesivir. Okay, remdesivir being quite interesting and so to say because it's a compound which was being tested for its effectiveness against SARS-CoV-2 in the study I will just introduce in a second. So let's go and do that. Clean up. I can start actually at two places, I can start here with compound, okay, in the landing page. I could do that. I will do something else just to introduce you to the fact that you have these such possibilities also inside the IDR. You don't have to hop and jump between the Omero web and this landing page. Instead, once you are inside here, you can click on compounds here. And now this doesn't give you an exhaustive list of compounds which are in IDR. That is not so. You have to start typing something. I will start typing remdesivir. And if I wait for a second, I typed quite a lot already. But you can see there are many, many compounds. And of course, the more I know, the easier I can narrow down. And then I accept the suggestion, and this already is finding out the remdesivir in 216 images of one study. And the number 3, it means that I have one screen under which there are 3 plates. In the meantime, you know already what plates are. This is a study from Philip Gribbon's lab, basically one of the first studies screening the, as I said, the effective compounds against SARS-CoV-2, trying to basically suppress the lethal activity of the SARS-CoV-2 on human cells in vitro. And there were quite a few compounds studied in this study. And let's just concentrate on the remdesivir, because that was, so to say, the most effective compounds found by the authors. If I expand the first plate and select the first field, and well, again, you can see the first the first nine images are all coming from one well, A10. Again, these are only the images associated with remdesivir here. Okay. Whereas the study obviously have many more images associated with all the other compounds which were being screened in this study. I select one of those images and go again to attributes. I can see that there is a compound name remdesivir. And if I click again on the icon, we know by now that this leads me to the outer world. And in this case, it's the PubChem database, where I can read the more particularities about this particular chemical. For example, its structure and, of course, molecular formula, and then some descriptions, etc., etc. And I am also noting that the authors submitted here a very nice analytical section under the which were curated and stored by the curators under the others section, which contains a parameter called percentage inhibition DPC, which in this case is 104.17. So what does that mean? Percentage inhibition is a parameter which shows how much does this particular compound, in our case we are talking of course about remdesivir, how much is this compound inhibiting the cytotoxic effects of the SARS-CoV-2. So I have my human cells here in a monolayer in a well. I put my SARS-CoV-2 on it, and then on top of that comes remdesivir. Now, the remdesivir either stops the SARS-CoV-2 from killing the human cells or not. Now, the percentage inhibition is quite high here, and as you can see that this stopped the remdesivir stopped the SARS-CoV-2 from killing the cells, because they are still nicely in a monolayer, and thus the thumbnail looks quite white. If I go here a little bit lower, let's say onto that thumbnail, which looks much more black, that's because the monolayer is much more sparse. There are many more dead cells. And I can see now here in the others that the percentage inhibition is much lower, as opposed to 104 I had in that very nice whitish monolayer. I have here 30.04 only. And this is now for what you can see, an example of what you can study and see in the manual user interface. If you are interested in going in more depth in that, we have also a Jupyter Notebook. I highlighted in my presentation what Jupyter Notebooks are. Together with a nice user-friendly environment, you can search and explore the effective dose response curves for all the compounds in the study. And you can plot the dose response curve for this one and find the most effective, the LC50 concentration, where basically the half of the cells are rescued. Okay, so this is quite a typical toxicology parameter. And I will just give you a hint, because now we are done with the user interface searches, and we are going into the searches which would be more elaborate by means of computational resources, but not by means of your effort putting into that, because Jean-Marie and the team put the effort there for you. So let's go for that. By this I mean that we will go and search using the API. Okay, so we are now at the point 4, search images in IDR based on metadata via API and notebooks. Okay, and what I was talking about being on that study just a second ago, which is the IDR 0094 from the screen for effective compounds against SARS-CoV-2. What I was talking about is the notebook which you can navigate to and study with me if you click on this link in your walkthrough. So you see that you are in the repository called IDR 0094 SARS-CoV-2. Don't have to worry too much about that. And the notebook is in plain English and it's very easy to read. And it's basically demonstrating how to explore the metadata associated to this paper. We know what the paper is now. It's the SARS-CoV study, the screening study from Philip Ribbon's lab. And you have the abstract here, etc., etc. And you can, let's say, accept all cookies, for example. Of course, then search for remdesivir. And, oh, sweet. Okay. And you can find here the remdesivir IC50, the IC50 being the effective concentration for stopping the cytotoxic effect of the SARS-CoV-2 down to 50% or reducing it down to 50%, and the effective concentration is 0.076, and then you can basically explore by launching this badge. I will not do that. You are free to do that, and this is then on your own time. Okay, as I said yesterday, the binder service is a free service, but you have to pay by your time and patience. It's usually quite fast. I definitely don't want to say that it's somehow bad, but yeah, if we all click on that now, we can have quite a wait. And you can also build the notebook locally on your machine if you choose so. And what it does is basically prepares some necessities first, and then loads the metadata and looks inside the metadata for the compound called remdesivir. The metadata, of course, being dragged from IDR into the notebook environment, and then filter by compound. Of course, unsurprisingly, I take the tables, filter for remdesivir, and then I calculate the half maximal inhibitory concentration from the metadata which are in the study. Okay, so I'm not reanalyzing the images, or this notebook doesn't reanalyze the images. It just takes the metadata which are there and calculates this plot, dose response curve, and comes up with a number which is then quite well within the range I just showed you in the paper. And it also plots for you examples of the range of different views of the cells, taken of course from the images inside the IDR. We know what are the concentrations of remdesivir put onto those images because we have the metadata from IDR, and we can indeed see in those images the dependencies I just talked to you briefly about in the UI itself, going from very little remdesivir, which really doesn't stop COVID from killing everything, and then going over more and more, adding more and more remdesivir, which then probably leads into some saturation effect and plateauing of the curve, as you can see here, maybe even resulting in then remdesivir itself having detrimental effects on the cells, which I will leave now by side. Also, of course, just a hypothesis. But you can see that this is the level of exploration you can perform here. These are, of course, the data in detail, and then you can export the results table as a CSV and have them locally. Okay, so this is the first example of the notebook. As I say, there's nothing stopping you really clicking on that badge and really doing the whole thing live, changing the parameters or even looking for another compound. What we have also...
Maybe one thing, just to slow down a bit. This is done in the multiple languages that you can use. If you remember the slide from yesterday, so if this is R, we use the R to access the data, but obviously, as Peter in the slide, you can use Python or Java to access the IDR data. So you're not limited to one language, programming language, for people wishing to do that. And one point probably to clarify because Alex was mentioning earlier on about the credential. In the case of IDR, when you do the UI, you won't need a credential. Sometimes, depending on the language, for that notebook you won't, Peter, you are not seeing, because you use the JSON API. But in some cases, you will find in notebooks some connection credential, and they're already in the notebook. It's public, so there's nothing to hide there. And it really depends on the mechanism you used in order to connect to IDR. Over.
Okay, thank you very much for this helpful comment. Yes, I will not now try to find the proper notebook with the public, but definitely there are different ways how this is done, and some of those notebooks, I mean, are of course in the walkthrough. Now, this maybe is something which I should show just to document. The various places where you can find the notebooks and start them from those places. Here under A1 is written, also linked from the IDR 0094 screen. IDR 0094 being the accession number of that study we are now belaboring. This is the SARS-CoV-2 screening study. And you can see that when I clicked on that link from the walkthrough, it led me to that screen B. Basically, that study, you can see, has kind of like a dummy top container. It doesn't even say screen, it just says SARS-CoV-2, Ellinger SARS-CoV-2 94. Then it says screen A. That's not the one I want, and then it says screen B. That's very common for many studies in IDR, something, a message to take home. Okay, this is one study, those three boxes here, and this is kind of like a header, and these are the two A and B, the two kind of chapters of that study, if you want. And I'm interested in the screen B here, okay, because that's where the juicy metadata are. And if I go now and expand the attributes on that screen B, and then go to the bottom, I have here a specific block, which you didn't find in the other studies. We are just starting with addition of those linkages into the Homero web of IDR itself. This is, so to say, early bird. And it says that study notebook is here. Again, the icon is for the outer world. The circles denote this interesting yellow, blue, red circles of binder, and I can start the notebook here. And this is what you can do. And now it's not just going through that notebook as I was doing on a GitHub, but in this case it went very quickly. Now that's live, okay? And yes, I can now go and click on the cells here and start basically executing if you do that, then please note that anytime when I click on a cell, there is first a number. I click on that, there was a star for a short moment, then the star turned into a number again. And that means that the cell is done and dusted. And I can go down to further cells. You can see the stars are persisting here. So this is the fetching, etc., etc. So I will leave you to play with that. And you can also go and study the Shiny app, which is again a link. I'm just linking straight from here, and it gives you basically a small user interface where you can switch between the compounds. I will leave it brewing here and maybe come back to that later, because there is no guarantee this will be very fast. Okay, and so just to make the point which we are trying to emphasize here is that if you are a submitter, then don't be shy to submit things like scripts and metadata of analytical results, which you might be thinking are very complex, but it doesn't matter. It matters only the quality of your annotation, basically. How well are you able to pass the information on the IDR team about what belongs to where and how it should work. Okay, and you can see on this example that if you do so, then the analytics will be exposed, if we of course manage to do that, but we will in that case of course make some effort to do that, as we can see the effort from the author. And then this gives you a possibility to explore the data further, or to give the scientific community the possibility to explore the data dynamically, meaning change the parameters inside the notebook and ask more questions, or even adjust the notebook themselves and compare the findings with the original analytical results, or use that as a quality control or a training set for themselves, etc., etc. Okay, now as this is still building, I will go to the case B, where we have yet another notebook, which is really just exploring metadata. I mean, the word is of course in a big inverted commas, which we call the name of the notebook is called Compound. Basically, the notebook is designed as follows. You have here a cell which you can use for defining a set of compounds which you are searching for. So this is basically a search many to many. If you happen to know what does that mean, meaning that you can have a list of compounds, and then you of course will be confronted with a list of images which are associated with those compounds across many different studies, as many as they are hitting matches for your list you submitted here. So nothing is stopping you to change, of course, those names of the compounds, find some other compounds. This will give you a table, and then you can explore the data in the table, determine the number of studies you have. Then you can start exploring the images which are in those studies and which images are associated with which compound. And you can see now again, it is some quite big column saying unknown. This is this is basically that the authors did not submit metadata for this particular image, which is always a pity. And then the notebook is basically giving you a possibility for creating an Homero figure out of those images. And although you can't expose the Homero figure on the IDR Homero server itself, because it's a read-only server, you can download the Homero figure, and if you happen to have the Homero, let's say, installed in your institution, or you have access to some other Homero server, then you can upload the figure into the figure interface, because as I said, the figure is a lightweight recipe, and it will fetch the images from the IDR and be available for you in that form. You can recognize the Omero figure user interface, which if you were on the workshop yesterday. Okay, so this would be the second case. What you could do with the Omero API. These are really just tips of the iceberg, okay, which I'm able to show from Jean-Marie's work on these possibilities of searching of IDR via API. And you can also have a notebook where you come to the IDR with a seriously prepared let's say search query, which comes from researching the outer world, so to say, you know, the databases, the other databases, let's say in this case, Intermine, which has a subsection called Human Mine. And if you do so, then you can basically use the notebook, which comes as an integrating package. Basically, the notebook is, and now let me switch back to my presentation because this is quite a juicy topic, so that we don't all drown in it. I have here a special slide for that. Okay, so this notebook basically starts by researching the human mind database. As I said, this is nothing to do with IDR to start with, but the notebook, the human mind also has an API, an application programming interface, and because the notebook is cleverly written, it can basically query both the human mind as well as IDR, okay, and then put those two together into a train of workflow, which makes quite a nice sense in the end. So first it queries human mind by passing the query for pancreas and diabetes as tissue and disease type, and this human mind answers with a set of genes, one of which is PDX1. This is done via, this is then put into the IDR multi-omics API, okay, and then in the IDR, as it says here, search for images in IDR associated to genes found in the human mind. Okay, you can simply take some or many or all of those genes found and then query IDR for images which are then ultimately, of course, associated with diabetes in pancreas. Okay, and just for an example, if we have from this list retrieved from human mind, just picked up the PDX one, then this is one example gene. And then the question can be, of course, narrowed down, give me just images linked to gene PDX1, and then the notebook returns those images with picking up on some interesting metadata as well and delivering them in one packet and parcel. Okay, now you would think, oh, this must have been jumping between many applications. No, what I'm trying to say here is that the notebook does all of that for you in one breath. Okay, you are still in the notebook, you can just edit the cells inside and it will do all this whole train for you, and in the end you will have an example for the PDX1 if you choose so. Of course, you can edit that. With the developmental stages, these are code words for the developmental stages of the gene associated with pancreas in diabetes in the IDR. They are associated with the study Accession 070, which was published by Kervin et al. In Journal of Anatomy. Okay, but that of course depends on what you will find, which depends again in turn on what you put into this very nice notebook. Again, easily launched by this star with this icon, and again, it's not. There's no guarantee that this will go very fast or some such So let's leave it in peace. But that's basically what I was describing on the slide. You are basically first querying for genes in human mind based on your first question, give me all genes associated with pancreas and diabetes, and then later find something, okay, and then pick something and then go with that into IDR, search for images in IDR associated to the genes found in human mind. And then the notebook will do, of course, all of that for you. You are just clicking run, or changing the parameters, clicking run. And then you can select subselect the genes, and it will give you back the images in the developmental stage, of course. So on the slide, they were, of course, only best of, but that's what the notebook does. I think I would like to end here. That would be, I just draw attention to the fact that you can build all the notebooks under B and C are actually in one big bunch under the repository called IDR Notebooks. The Shiny app with the curves, look, it's here. So let's just try to explore. These are the different curves of the compounds of the SARS-CoV-2, where the effective compounds against SARS-CoV-2 were investigated, and this is the Shiny app, which gives you quite nice dose response curve for every compound. There are 333 compounds, so it's not by any means somehow minimalistic what you have here. And this nice little UI built again very quickly by one click of the button. Thanks a lot for your attention.